The next speaker will be Ryan Chae with a presentation on Parkinson's disease.
Hello, my name is Ryan. I'm a sophomore at GIDAS, and I'll be introducing about Parkinson's disease. So first of all, before I begin to explain what Parkinson's disease is, Parkinson's disease in the United States, about 1 million people in the population of the US are diagnosed with Parkinson's disease. And first of all, I'll briefly explain about Parkinson's disease. Parkinson's disease is a brain disorder which causes uncontrollable muscle movements such as symptoms and shaking, stiffness, and difficulty body coordination. If you see on the image left, there are more common symptoms caused by the Parkinson's disease. The main causes of the Parkinson's disease would be the degeneration of nerve cells in substantia nigra and the lack of dopamine. And substantia nigra is a basal ganglia which controls the movement. During my formal experiment, I hypothesized that several genes are either overexpressed or underexpressed, which induce the Parkinson's disease. The method I used to conduct the experiment was through the GEO research study and the analysis of graph and statistics. This is one of the data I used for my experiment. As you can see on the image right, this represents a volcano plot. The cases that are represented as blue colors are the underexpressed RNA, where the cases shown in the red dots are the overexpressed RNA. As the cases are plotted more higher in the graph, it represents how the cases are more significant. And one example of the underexpressed RNA, which was significant, was the HSA-MIR 51895P. This underexpressed RNA mimics the indigenous miRNA and enables miRNA functional analysis by upregulation of the miRNA activity. This therefore shows how miRNA activity in Parkinson's disease decreased. Another example of the underexpressed RNA was the HSA-MIR 8084. This underexpressed RNA promotes the proliferation and the DNA synthesis. These RNA can be assumed to suppress DNA synthesis in the Parkinson's disease therefore. An example of the overexpressed RNA which was significant was the HSA-MIR 432. This overexpressed RNA is a microRNA that has been implicated in various biological processes and diseases. This shows how the correlation between the overexpressed RNA and the Parkinson's disease exists. Another example of the overexpressed RNA was the HSA-MIR 3913-5P. This overexpressed RNA mimics the indigenous miRNA and enables miRNA functional analysis by upregulation. This therefore increase in miRNA activity which leads to Parkinson's disease. These are two more graph analyses that I conducted in my research. The graph on the left compares the cases between the overexpressed and underexpressed RNA. As the cases are plotted more higher, it represents how the cases are more significant, and as the cases are plotted more right, it represents how the RNA is more overexpressed. The graph on the left represents each individual with either the Parkinson's disease or people without Parkinson's disease. The green box represents the individual with Parkinson's disease, and the purple box will represent the individual without the Parkinson's disease. The graph without any noticeable outlier represents how this graph was consistent and did not contain any significant outlier. To conclude my experiment, I stated how underexpressed RNA, such as HSA-MIR 5189-5P and HSA-MIR 8084, and overexpressed RNA, such as HSA-MIR 422 and HSA-MIR 39135P, reduce the Parkinson's disease. This was further supported through the GEO graph analysis. Thank you.